Hi guys, this is Ayan Parichha and you are watching my YouTube channel. In this video, we will learn about the concept of non-cyclic photophosphorylation, also known as Z-scheme or Z-scheme. So let's get going. Whenever you have gone through the chapter of photosynthesis, I am sure that you have come across this specific diagram. Now this specific diagram depicts the non-cyclic photophosphorylation. Now let's understand basically what this diagram tries to tell us. Now this diagram tells that when sun ray falls on PS1, then one electron gets excited and jumps out of PS1. That electron is then taken up by NADP, which eventually reduces NADP into NADPH. At the same point of time, when sun ray falls on PS2, another electron gets excited and jumps out of PS2, and that electron moves through an array of molecules and finally goes to PS1 to meet up the electron deficiency which was caused due to capturing of that electron jumping out of PS1 by NADP. Now this array of molecules through which the electron passes is known as the electron transfer system, or in short the ETS. Now at this point of time, as the electron jumping out of PS2 was not returned to PS2, electron deficiency is created in PS2. Now this electron deficiency is met up by lysis of water molecules to give oxygen and hydrogen, and in this process the released electron is taken up by PS2 to meet up its electron deficiency. Now this lysis of water in presence of sunlight is known as photolysis of water, and the oxygen molecule released the oxygen molecule thus released is the byproduct of photosynthesis that you see in the equation. Meanwhile, when the electron jumping out of PS2 travels to PS1 through the electron transfer system, some amount of its energy is taken away by an ADP molecule which gets phosphorylated into ATP as a process of electron moving through the electron transfer system. Now by this process, the two high energy molecules of light reaction of photosynthesis, that is NADPH and ATP, are synthesized, and at the same point of time one of the major byproducts of photosynthesis, that is oxygen, is also produced. Now you might be wondering why electrons are jumping as if they are jumping in a high jump competition. Is it so that the electrons are going to participate in some Olympic event? The answer is no. So why they are jumping like this? So let's try to answer that question. First of all, we have to understand that this diagram is plotted on a graph. So if we take up a graph sheet and we draw the axis like this, and on the y-axis we take reduction potential, whereas on the x-axis we take space. Now the space of x-axis simply represents the spatial arrangements of the molecules participating in this non-cyclic photophosphorylation. Now what we have to learn is what is reduction potential. Don't worry, I am coming to that in a moment. But for now, let's consider reduction potential to be a quantity. And now considering that, you can see the electrons after jumping out of their molecules have certain reduction potential, and then the electron transfer system has lower reduction potential as compared to the electrons, and then comes the reduction potential of PS1, and finally in the lowest part comes the reduction potential of PS2. Now the question that is coming to your mind is, what is reduction potential? To answer your question, we need to know two things. One, the idea of reduction reaction, and two, the idea of reduction potential. Now let's come to the reduction reaction first. Now I hope you all have learned chemistry at some point of time in your academic life, and there you have learned that reduction can be explained in two ways. First, by addition of hydrogen, the classical theory, and by addition of electron, that is the electronic theory. So basically over here both the reactions that I have show the oxidation process. To understand reduction, let's first understand the concept of oxidation. So according to the classical concept, oxidation means removal of hydrogen or hydrogen-like molecules from one molecule. So if hydrogen is removed from AH2, then we can say that AH2 has been oxidized into A. The electronic concept says if electron is removed from a certain molecule, then that molecule is said to be oxidized. Now, in the second reaction that we can see, we clearly understand that B is undergoing oxidation and it is getting oxidized into B positive monovalent ion. Similarly, the opposite means reduction. That means addition of hydrogen or hydrogen-like molecules to certain molecules signifies reduction according to the classical concept of oxidation reduction, and addition of electron to certain molecules signifies reduction according to the electronic concept of oxidation and reduction. Now with this clear concept of reduction reaction, now we move on to the next part of our understanding, that is understanding of reduction potential. Now to understand reduction potential, let me bring in one nice analogy. In our childhood, we had mainly two jobs in our life. First one is to study, which we all disliked, and the second one was to play. But there were certain students in our class who used to love studying, and there were other students in our class like me. Now if I say a particular student has a higher studying potential, that simply means that the student has higher tendency to study than the tendency to play. At the same point of time, if I say one particular student has higher playing potential, then that simply signifies that the student has a higher tendency to play around rather than studying. So similarly, if I say that one molecule has higher reduction potential, that simply signifies that the molecule has a higher tendency of getting reduced, or it wants to get reduced readily. Right? So I hope this clears our idea of reduction potential. Now, equipped with the knowledge of reduction potential and the concept of oxidation reduction, we can move on into the actual process of Z-scheme. To understand Z-scheme, we have to understand where Z-scheme takes place. Z-scheme takes place on the membrane of thylakoid, a structure present within the chloroplast of plant cells. Now, so let's take up a membrane now, and let's assume that this membrane is the thylakoid membrane. So outside of this is known as the stroma of the chloroplast, and inside the membrane is your thylakoid lumen compartment. Now let's bring in the other molecules that are present in thylakoid lumen and in the stroma. So let's start with the stroma. In stroma, we have NADP, that is nicotinamide adenine dinucleotide phosphate, and we have ADP, that is adenosine diphosphate, and along with that we have protons. And we have other molecules which are not significant to this discussion. In the thylakoid lumen compartment, on the other hand, we have protons, but no other significant molecules except water. Now, one thing that you can clearly see is that the concentration of proton is evidently higher in the stroma than in the thylakoid lumen. Now let's bring in the molecules playing direct role in the non-cyclic photophosphorylation. Now first comes our PS1. Along with that comes one enzyme, that is NADP reductase. Next comes PS2. Then comes a molecule known as plastoquinone. Then comes another molecule known as cytochrome B6F, and finally comes a molecule known as plastocyanine. This plastoquinone, cytochrome B6F and plastocyanine together make the electron transfer system. And finally, we have another very important molecule, that is FOF1 ATPase. One thing I want you to remember very carefully is that this molecule is not F0F1 ATPase. It is a mistake commonly done by students as well as many educators. It is not F0, it is FO. Now if you want a detailed video on that, please let me know in the comment section. if I get enough response, I will make one video dedicated to FOF1 ATPase. Now, as we have got introduced to all the molecules and structures which are directly involved in this Z scheme, in this non-cyclic photophosphorylation, let's begin with the process. Now, far away from the plant in the sky, somewhere we have the sun. Now, sun emits its radiant energy in form of sun rays. Now, sun ray simultaneously fall on PS1 and PS2. So, let's see what happens when sun ray falls on PS1. As soon as sun ray falls on PS1, one electron gets excited, and that electron is taken up by NADP reductase, and this NADP reductase enzyme transfers its electron to NADP and forming NADPH or reduced NADP. Now, from reduction potential point of view, if we discuss, then we can clearly understand that when the electron in PS1 got excited, in the vicinity of PS1 there was only NADP which had a higher reduction potential, and that's why the electron moved to NADP. Because NADP has a higher reduction potential, it has a high tendency of getting reduced, and that's why it took up the electron, and this process was facilitated by NADP reductase. Now you can clearly reason the name of the enzyme, since receiving electrons or addition of electrons to a molecule signifies reduction. That's why the enzyme which is adding electron to certain substance is obviously known as a reductase. Now this creates an electron deficiency in PS1. Now sun ray at the same point of time falls on PS2, and similarly one electron, as they said, jumped out of PS2 or got excited and then got taken up by the plastoquinone. To meet up the electron deficiency in PS2, water molecules present in the thylakoid lumen compartment got lysed into hydrogen and oxygen. Now this oxygen that you can see is the same oxygen that you see in the photosynthesis equations as the major byproduct of photosynthesis. And in this lysis process, the electrons evolved got taken up by the PS2 to meet up the electron deficiency created over there. Now you may ask why PS2 could not take back its own electron and has to meet up its electron demand from breakage or lysis of water. Now the answer is very simple. When the electron got excited in PS2, in the near vicinity there was only one molecule, which was plastoquinone, which had higher reduction potential than PS2, and that is why it took away the electron from PS2, and then it got transferred to cytochrome B6F. Right. You can go back in the video and can refer to the graph showing reduction potential of the different molecules. Now the electron from plastoquinone moved to cytochrome B6F, and from cytochrome B6F this electron moved to plastocyanine, and this movement of electron through this electron transfer system pumped protons through cytochrome B6F into the thylakoid lumen. Thus, you should always remember that cytochrome B6F is a proton pump, which pumps protons from stromal compartment into the thylakoid lumen compartment. Right. Now, the electron is with plastocyanine. Now, since PS1 has higher reduction potential than plastocyanine, it will readily take away the electron from plastocyanine to satisfy or to meet up its electron deficiency, and thus the electron deficiency of PS1 will get meet up, and the electron deficiency of PS2 will also get meet up in this process. And what will be the consequence of this? In the consequence of this is that the NADP molecule got converted into or got reduced into NADPH molecule, and a lot of proton got pumped into the thylakoid lumen. As this process will keep on continuing, after some time there will be a high proton concentration in the thylakoid lumen compared to that of the stromal lumen. This will give rise to a concentration gradient of proton having the higher end in the thylakoid lumen and the lower end in the stroma. Now this concentration gradient will be used up by the FO-F1 ATPase, and the protons will be channelized along the concentration gradient, and this channelization of protons will lead to phosphorylation of ADP present in the stromal compartment into ATP. Now at this point we can clearly see that both the high energy intermediates of photosynthesis, which are synthesized in the light reaction, that is ATP and NADPH, are synthesized. These two are essential for dark reaction to occur. At the same point of time, oxygen is synthesized in the thylakoid lumen, which is the major byproduct of photosynthesis. So this is in brief the idea of non-cyclic photophosphorylation. Now this is a concept map of non-cyclic photophosphorylation. Now if you want this concept map, it is available on my Instagram page at Ayan_biology. Follow me on my Instagram for concept maps, flashcards, video flashcards and quizzes. Now the question that remained, why this non-cyclic photophosphorylation is known as Z scheme or Z scheme. To answer that question, let's rotate this diagram 90 degrees. Now once we have rotated the diagram, now if we trace the pathway of electron movement in this diagram, then we can see it closely or roughly resembles the structure of a Z or a Z, and that's why this non-cyclic photophosphorylation is said to be a Z scheme or Z scheme. Now I hope you clearly understand what exactly happens in non-cyclic photophosphorylation and why it is known as Z scheme or Z scheme. Thanks for watching this video. If you enjoyed this video, please hit the like button, share this video among your peers, and subscribe to my channel for more useful content, and finally press the bell icon and click the all button to become a part of our notification squad.